And we're good.
All right. Okay. Hi, everyone, and welcome. I'm Fatima Osman, and together with Irene Lavaji-Craddock, we lead the NCPN Quality Initiative. Today, we're going to take you through a quick refresher on quality management, what it is and why it matters, and how we all can keep improving the work we all do every day. This session is part of our ongoing commitment to maintain a strong and consistent quality management system, or QMS, across NCPN. We really promise that this won't be a lecture full of acronyms and checklists and very painful, but our goal is to make QMS feel relevant, practical, and even a little bit inspiring. So let's get started. Oops, no. Sorry, it's acting up. So we've made significant strides in ensuring that the quality initiative remains responsive to the needs of various centers across the commodities. So to achieve this, we've established a continuous feedback mechanism through surveys and think tank events to ensure the quality initiative always remain guided by the real needs of various centers across the commodities, because it's shaping its activities and products. So since its launch in 2019, the NCPN Quality Initiative has made significant progress, starting with 2019, where we had our first quality workshop and established a steering committee. A year later, we have started developing the NCPN-centric standards and had a couple of training to educate ourselves and educate the network on QMS aspects. In 2022, we have completed the final draft of standards, but also wanted to distribute a survey to get a feel of the real need of the network to establish the direction that we're heading. In year 4, we started implementing the request expressed in the survey by creating an SOP and positive control shared Google list. In year 5, we again, as requested in the survey, we have offered trainings on qPCR and high-throughput sequencing. Recently, we have conducted a think tank workshop to establish a continuous feedback mechanism, ensure again that we remain responsive to the need of the various centers. So the survey, along with the think tank outcomes that Irene will discuss later, served as our guiding compass to shape the future direction of the initiative. And your contributions have been really valuable in shaping and establishing this initiative. The NCPN Quality Steering Committee developed the survey criteria for diagnostics, therapy, foundation, and administration in order to gather vital quality management information. The survey consisted of 5 sections: the background, center activities, clean plant training, quality processes, and additional questions and comments. I'd like to share the key findings from our survey, which helped us shape this quality initiative. First, we wanted to have a statistical feel of what have centers done so far to adopt QMS. For example, do they have a lab documentation system in place? As you can see here, 25% have lab documentation systems, others have LIMS, but the majority use conventional Excel and MS Access. The survey also inquired about areas where centers believe their programs could benefit from quality principle. Almost all quality management system concepts were desired through this survey, including document tracking, data management, training documentation, among others. In terms of training, we wanted to know which areas centers prioritize for their training. The sequence of training areas that are desired was diagnostic methods, plant production and propagation, plant distribution, and therapeutics. We have also conducted a mini survey in 2023 to keep checking on the most important training and see if anything has changed. Centers have requested data management refresher workshop, and this is exactly what we're offering right now. together with training on diagnostics, plant production, introduction, and therapeutics. Furthermore, we wanted to identify what specific trainings on techniques and procedure would be useful to the centers. These 4 aspects, as you can see here, were repeatedly requested, which is virus elimination, high-throughput sequencing, qPCR, production, distribution, and data management, as well as SOPs. So we therefore have offered the workshop on qPCR and high-throughput sequencing back in 2023, and created also an SOP shared Google list. And next on the list will be the plant production and distribution, and as well as therapeutics. So we aimed also to investigate whether NCPN centers maintained an SOPs and work instruction for their general activities. The responses revealed that centers have SOPs for all diagnostics, others have some, others have few, and some of them have none at all. So we also asked centers if they are willing to share positive controls with others. The responses were overwhelmingly positive. Therefore, we started a shared Google document, as I mentioned, for the SOPs across the 7 commodities. It is a voluntary aspect, as well as another voluntary shared Google site where centers can also collaborate and share their positive control lists. So the survey included also questions on sample tracking from introduction to distribution to sample retesting. Almost 81% track manually, others use LIMS, GRIN, and some barcoding and other tools like, as I mentioned, Excel sheet. The tracking distribution data to customers is primarily done manually by the majority of centers, as you can see from this survey questions. However, a small percentage of centers utilize more advanced systems, such as 15 employing the LIMS and GRIN, and 31% use systems like ADAPT and local database, such as Excel. So the discrepancy highlights a need for standardizing training on distribution procedure, including tracking from production to distribution, which is likely to become a priority in the near future that we offer the training for. Thank you, and I'm done with, Irene, I'm done with the survey. If we'll keep the questions at the end.
Okay. All right. Can you see the slides?
Yes, we can. Yes, we can.
All right, so you just heard the results from the surveys that were conducted in the past, and now I'm going to give you a little bit about the think tank event that several of you attended back in June. And before we dive into quality concepts, we wanted to really take a moment to reflect on this NCPN think tank event that was held just this past June. It was a focused event that brought together NCPN leadership, industry partners, scientists, and program managers from across commodities. The goal was really to take a step back, re-examine our shared mission, and reimagine what a resilient, effective clean plant network should look like moving forward. So again, you know, it's a quality, not just isn't a check in a box, but quality in the broader sense, how we maintain that trust, transparency, and excellence across the programs, while also being adaptable. And today's refresher is really part of that momentum. It's a small piece, but it's an important piece in the bigger picture of how we keep growing together as a network. So at the think tank event, we took time to reflect not just on where the Clean Plant Network is today, but where it wants to go. The major takeaway was that quality and sustainability go hand in hand, and we can't really talk about long-term success without showing that we have reliable, transparent systems in place. And so several priorities emerged at the event, which included strengthening communication and coordination across the network, aligning quality efforts across centers, and building that flexibility that is needed in our systems while still maintaining accountability. There was also a strong push to really reduce the burden of documentation where it does not add value, while still keeping the core quality practices front and center. Today's refresher is again something that was mentioned during that event, and something that falls in the remit of quality overall. And so we thought that it was a time for a refresher that could also be shared and reflects more the views of the current NCPN network. So following the think tank, these were identified as key priorities for the NCPN centers, the things we need to focus on to keep the network strong and aligned. And the critical needs that were identified were critical training needs, there were data management needs, and there were enhanced collaboration needs. And so these priorities aren't really specific to individual centers. They're about aligning across the entire network. And again, today's session is part of that effort to make sure that we're all on the same page when it comes to quality, and to give everybody the tools that make it easier in your day-to-day work. So now the big question is really, how do we turn all these great ideas into action? Well, the think tank gave us a strong foundation, but the implementation happens at the center level in the day-to-day work that we all do. And that's really where quality comes in. If we want to build trust, that demonstrate the value and ensure long-term sustainability for the network, then we need systems that are clear, consistent, and effective all year long. What we're about to walk through now is going to be a quick refresher on the core elements of quality management, not so much just as theory, but as a practical tool to help you translate those big picture goals into meaningful habits and improvements. And so with this, we're going to transition to the next part of the agenda, really what is supposed to be the bread and butter of quality and quality management, and the refresher. So I'm going to give a brief overview to understand what quality management is, its importance and implementation. We're going to have a definition, and then we're going to look at some key actions that are included. All right, so let's kick off things with the simple question. What do a Hershey bar and a great lab test result have in common? The answer is consistency. No matter who makes it or when, the result should be the same. So that's what a quality management system, really, or abbreviated QMS, sometimes can help us achieve. It's not about red tape. Quality management is not about making things difficult. It's about reliable processes, trusted outcomes, and always looking for ways to improve. Over the next few minutes, we'll break down what a quality management system is and how it supports and can support everything we do, from the lab test to the documentation and customer satisfaction. So what exactly is a quality management system? The definition is that it's a coordinated system that helps us manage quality in everything we do. And that includes documentation, training, problem solving, and most importantly, improvement over time. It's not just a manual, a binder on a shelf. It's really the living system that you create and you operate under, that supports that consistency, transparency, and accountability to ensure those great outcomes again and again. There are 4 pillars of quality and a quality management system. There are lots of different quality management systems, from the most restrictive to the most demanding. You can think about something like ISO, or the ones that are used by different doctors and different associations. But really, it comes down to 4 key elements. And those are that you say what you do what you say, you prove it, and you improve it. And when we follow this cycle, we create processes that are clear, repeatable, and always improving. And that's how we build the trust and avoid the not so good surprises. So the first step is that we said is that you say what you do. That means you're documenting your processes in a clear and useful way. So the policies and procedures, the instructions, the records, SOPs, it should all be written in a format that your team can actually understand and use. And if it matters to quality, it should be documented, not just for sort of internal audits that you may want to run in your lab every now and then, but really for consistency of your processes. The next step is you do what you say. So this is really about aligning the practice with process. So you can write the perfect SOP, but if you don't follow it, you're not really managing quality. And that, so if what's happening at the bench doesn't really match what's on paper, then we have a gap. And that's where the problems can begin, and where a quality management system can help bring things back into alignment. After you've done what you said you were going to do, you need to prove that you've done what you said. So we prove that our system works through evidence, things like records, little internal checks that you can do, internal audits, or the sort of feedback that you get from your customers, and more thorough internal reviews that you may want to organize. So you can think of the little checks that you do on a regular basis of how things operate as a dental checkup. They help us catch small issues early before they become bigger problems. And once you approach it as this is not about assigning blame or pointing the finger at someone. It's about improving the process. Once you put it on a schedule, rather than waiting for a big problem to emerge and then trying to solve it, then a more positive experience can be created for everyone. The final step is the improvement. So a quality management system is not just about keeping things stable, actually. It's about solving problems and preventing them from recurring. One example that can be taken is the 2023 Taylor Swift Ticketmaster crash. So the quick fix was to pause the sales and communicate with users. And then a corrective action was put in place later when they improved the system capacity, updated the algorithms, and changed how high demanding events can be handled. So for that, a root cause analysis had to be in place, had to be implemented. And that's what these quality systems are built to support. So you can make it more complicated and then divide into what are called preventive actions. So it's something before the problem emerges that you are putting a measure in place, right, to prevent something bad from happening, or something bad happened and now you have a corrective action. Regardless, you're trying to find ways to improve your system in a preventive or in a corrective way. And to do that, you need to analyze, you need to do that kind of root cause analysis. Okay, one important point that I think is it's good to think about is that, as we said, the quality management system is not about keeping things stable, keeping them the same. It's about improvement. And at the same time, I think it's about also accepting risk, but managing the risk and including flexibility in your system. So risk management is about thinking ahead, right? What might go wrong and what happens if it does? The truth is we can't eliminate all risks, but we can prioritize on the ones that really matter and plan for them or do the best we can. So let's say, for example, my internet connection drops, which does happen at this facility every now and then I have a backup. My backup was to make sure that I have a phone that I can use as a hotspot. My slides have been downloaded by someone else, and someone else can carry on with the presentation. So the same thing really applies to lab work. A good quality system cannot be too rigid, but needs to be responsive. All right. A quality management system a quality management in general needs to be part of the culture of the place we work at, of our centers. Cannot be something that just shows up during maybe a big external review that you're having of your center. It should really be part of how we work every day. And that means open communication, celebrating small improvements and making sure everyone feels part of the process. it's really about creating that culture of consistency and continuous improvement. And finally, let's go back 500 years to Leonardo da Vinci. So he was not just an artist, he was a scientist, an engineer, a process thinker who deeply understood the value of observation, documentation, and iteration. And he documented every step of his ideas, inventions in his famous notebooks, where he recorded every step of his experiments and studies, sketched mechanisms in different layers, and noted the causes of failures and revisited the designs over the years. He was studying what worked, what didn't work, what failed, and most importantly also why. Why was something working or not working, not just the how. So Leonardo really is a great example of having applied root cause analysis to everything that he was doing. Long before we called it root cause analysis, and his commitment to precision, improvement, documentation laid the foundation for engineering practices that we're still relying on today. He believed in assigning tasks to the most competent individuals, depending on what the task was, and in understanding why something worked and not just the how, as we said. And Leonardo is a great example, I think, because, well, not just because he was Italian, but because he really lived the quality mindset to observe, document, learn, and improve. And that's a mindset that we need to bring to our work in the NCPN. So the final message I'd like to leave you with is that quality is not a modern invention. It's really a mindset. And it's the one that Leonardo da Vinci lived 500 years ago. So we can definitely do that with our work. And with this, I will give it back to Fatima.
Thanks, Irene. You've just heard from Irene about the general principles of quality management, but how do these principles apply to the operations of NCPN centers in our case? So the Quality Initiative is a platform for exchanging best practices to support centers in delivering high quality plant material to customers. So this is, in our case, is achieved through standardization, including creating and distributing NCPN-centric standards, collection of crop-specific procedures and protocols, and creating a list of crop-specific positive control. Well, a diverse working group consisting of members from 7 NCPN commodities, regulators, lab personnel, and quality management experts, as well as representation from larger and smaller NCPN centers, USDA APHIS, and NCPN management. They all collaborated to develop this NCPN-centric standard. Through virtual collaboration, the committee evaluated, drafted, and refined the standards, ensuring they were voluntary, realistic, and centered on delivering high-quality products. The group completed the first draft, which was then submitted to the Quality Steering Committee for review and approval. The NCPN-centric standards were then finalized and distributed network-wide in June 2022. What are the NCPN-centric standards? Those are, as Irene was mentioning, those are a set of requirements to help formulate coordinated activities directing the quality of the product. The working group used the principle of the quality wheel in drafting the standards, and the quality wheel has different components that if implemented correctly, then you reach the quality in your different activities. They have, as you see, components of the technical requirements, such as specimen handling, testing methods, administrative requirements as personnel budget, and then management requirements, such as reviews, internal audits, and complaint handling. Learning all these components of the quality wheel and the quality management concept led us to draft our own NCPN-centric standards, which were meticulously crafted to cater to the unique needs of the 7 NCPN commodities, covering a broad spectrum of activities. It was developed over really tedious 2 years. These standards provide a comprehensive framework and help NCPN members and stakeholders to meet their highest quality benchmark, totally aligning with the NCPN mission. They, as you can see here, they include, again, administrative requirement, management requirement, technical requirement. I'm going to show you some examples of what exists in the standard record or document. The example from the management requirement section of the standard is, for example, the document control, stating that NCPN centers must have documented procedure or work instructions that outline how centers document, control, and manage their work instructions or the procedure. These procedures or work instructions should include specific details such as document ID, title, authorization date, as well as amendment and revision dates. Another example is the records section in the management requirement. Records may be in the form of various types of media, such as hard copy or electronic media. In the case of computer-collected data, for example, similar efforts have to be done to prevent the loss or change of the original data. And another example is that in the technical section comes the testing and validation, which is very important for the diagnostic aspects of NCPN mission. It states that plant diagnostics, therapy, and plant production and maintenance processes must all be performed by authorized, qualified personnel using the current version of their SUPs or work instructions. These personnel must approve these procedures on a regular basis. Another example of technical requirement concerns the reference material. Where possible, NCPN Center use these reference material and calibration standards that are traceable to international standards. And efforts should be considered so that the biological reference material are traceable and accepted standards are established. The section regulates the sample and sample handling. The NCPN Center provide guidelines for collecting and shipping plant material to the customer. The centers must also outline the procedure for receiving, handling, storing, and depositing of plant material. The plant material must have a system for identifying to prevent the confusion. The identification is retained through its life cycle and linked to a final report. Procedure use must protect the integrity of the plant material. Another aspect of implementing NCPN requirements and standards is also through increased harmonization of crop-specific SOPs and creation of the positive control list for the NCPN commodities. Again, those two aspects have been requested by the centers when the survey has been conducted. We always wanted to keep in touch with the real needs of the various centers. As shown here, we discussed previously in the survey, the responses revealed that some centers having SOPs, others have some, few, or none at all. Consequently, we have developed a shared Google Hub for all the SOPs across the 7 commodities to voluntarily add their SOPs, which will be represented in the following slides. For example, this is a shared Google-based hub that the Quality Initiative has created, where the protocols and the procedure for all the crops can be stored. Those are the example, those are example for the procedure for several NCPN commodities. Like here, the NCPN grapes and NCPN citrus. We also have collected protocols and procedures from different centers within the NCPN citrus, within the same centers of the same commodity. For example, in this case, in NCPN citrus, we have collected from Texas, Florida, and California. Again, keeping in touch with the survey, the quality survey revealed an overwhelming support for creating and exchanging a positive control list. For example, this is an example, creating an NCPN citrus reference and positive control list, compiling a list of sources for positive control material for all types of pathogen that we use, for example, in viruses, viroids, and prokaryotes, which can be obtained from national institutions, as you can see from the right-hand side, the sources, or international institutions. We also created a list of an NCPN centers to provide real-time updates. We're informing centers within the same commodities about the availability of positive controls and also the format, how the positive control exists, whether it's in planta or as a nucleic acid, in case of viruses, viroids, and prokaryotes in our case. So the primary goal of these lists is to facilitate the collaboration among centers and enabling them to access all these information through a hub created by the NCPN Quality Initiative. Thank you. So let's continue with the agenda. Irene?
Yes.
Yes.
Can you hear me?
Yes, we can.
Okay, so the next speaker is Georgios Vidulakis with the MIKE guidelines. We're going to move on to the portion of the diagnostics.
Hello, everyone. Let me set up my computer here 1 second. Okay, share the screen.
While Georgios prepares, yeah, so the first portion of the workshop was a refresh. It was about reminding you of the previous events with the survey, the think tank, where we come from. Then we've had a refresh on the basic concepts, and now we're shifting gears, and we want to see the effect of quality on the operations of our center. So we're going to have different examples from diagnostics, therapy, and then maintenance and distributions, which are the functions of our centers. Thank you.
Can you see the full display screen? Yeah.
Yes.
Yes, we can.
And I cannot see all of you right now on my screen, all the participants, but I suspect that this is not the first time that you hear this term, the MIKE guideline. So just really quickly throw a digital raise of hand if you have heard this term before, because I think it's very critical for all of us that we work on molecular diagnostics to know about this. So the acronym MIKE stands for Minimum Information for Publication of Quantitative Real-Time PCR Experiments. See if we can advance from here. Okay, this works. The concept and the idea came in 2009 by Bastin and co-authors when they realized that the experiments reported using qPCR data was, the reports were inconsistent. They didn't have enough details, and that was leading to wasted resources, either from people trying to replicate the experiments or trying to implement the qPCR assay or experiment that was published. So you can see here the first page of that classic publication that all of you should have in your labs. And what they offered with this paper is a framework that assures the accuracy and reliability of qPCR, reported qPCR experiments.
Sorry,
Yeah.
Georgios. Sorry to interrupt you, but are you advancing the slides?
No, I'm still talking about this slide.
Oh, okay.
So the MIKE guidelines, they are very specific. They talk about they advise the authors on how to design their experiments, how to perform their experiment, and how to interpret the results from qPCR experiments. And they go in very a deep level of detail about how the samples need to be described or what kind of samples needs to be used, primers, probes, how the nucleic acids are extracted, how the how the qPCR conditions need to be reported, and so on. As an example, I wanted to let you know that we do our best at the CCPP, at the Citrus Colon Protection Program. We do our best to adhere to these guidelines. It's an evolution. Our original diagnostic papers, maybe they had a couple of elements of this, but as we are growing and we're studying and we're learning, we try to follow the guidelines to the biggest extent possible. And the most recent example of how we do this is this paper in Phytofrontiers in 2023 from Osman et al. From our group, where we took the very important assay to detect the Candidatus liberibacter species that are associated with the Huang-Longbing disease. We took this assay that was published in 2006 or developed in 2006. And I don't know if you remember, but the MIKE guidelines were published in 2009. So when this very critical assay was developed, there was no guidelines in place. So what we did was to take this assay, update it with back in 2006, the epidemic of HLB was just started, 2005 in Florida, so the genetic information on the bacteria was very limited. But 20 years later, there was much more genetic information on this bacteria. We had the MIKE guidelines in place. So we took this opportunity to put this paper together in the special issue of Phytofrontiers on Diagnostics. And I'm going to use this paper throughout this presentation to show you how we do our best to implement the MIKE guidelines for our assays. This is the core of the MIKE paper. This table 1 sits in the center of the paper, and it has general titles or general categories that are listed on the left-hand side of your slide, 1 through 6. And then each of those categories have essentially a checklist underneath. And, the major category, the experiment design, sample preparation, primer probe design, the qPCR protocol, how you analyze your data and validation methods. And just to zoom in a little bit inside the table, just as an example, you see the checklist, the different check-in items for each of those important categories for qPCR experiments. And what the authors did from Bastin et al. Is to flag some of these items with E for essential. You shouldn't even proceed with your experiments, and you shouldn't even submit your paper unless if you have those elements. And D for desirable, that if possible, you should include them in your study. And us as members of the NCPN and striving for quality, and especially on diagnostics, we play a dual role. As authors, we need to keep in mind those guidelines and make sure our submitted papers are fulfilling them. And also as reviewers, we need to make sure we keep the standard overall in the literature and send submitted manuscripts back to the authors and educate them and tell them, your experiment lacks these elements and so on. A paper that was published a little bit later from 2009 publication on MIKE on quantitative qPCR was from Broders and al., where they did something very interesting for us, which I think it fits best what we do in our centers, because in our centers we not necessarily care about quantitation of the target. We don't really need to know in most of our clean plant centers how much virus I have. We're more interested in a yes or no answer. So I found this approach, this paper that was, and this work that was performed to identify gene copies on genetically modified organisms, and they took the approach of what they call qualitative mice or mice for qualitative PCR. Is my target, in this case, is the transgene present, yes or no, in my sample? Which is very similar to what we're doing. Is the pathogen, is the virus, is the bacterium present, yes or no, in my germplasm stock on my sample? So what this publication did was to, and you can see on the table, they are comparing side by side what would be essential for a qualitative validation process or publication versus quantitative. And we have found in our lab this publication also very useful, and we are using the two set of guidelines in conjunction, in combination. So I would like you to know about this resource and use it as you see fit in your labs. To dig a little bit more into the guidelines, especially on the validation part, which is uniform across if it's qualitative or quantitative, the big 6 steps or big 6 elements of a validation process according to the MIKE guidelines is to identify and characterize the amplification efficiency of your reaction, the classic E equation there at the bottom of point 1, the analytical sensitivity or limit of detection, specificity, you're getting false positives, false negatives, robustness, how robust is your assay? If someone pipettes the wrong amount of reagents or uses the wrong software in the thermocycler and so on. Transferability, are you the only lab that can perform this assay, or other labs can perform it with exactly the same results? And reproducibility, if I run the assay today or tomorrow, or using this type of plates or these types of plates the other day or a different machine and so on, are my results going to be reproducible? And we don't have much time today to go through in detail all these. I think most of you understand the importance of those elements, but just a few examples. The amplification efficiency is one of the most important elements of validating a qPCR assay. Again, this is the example of our recent publication. You want the series dilutions that are used to calculate the efficiency. You want them to have a linear relationship with each other. You want this every tenfold dilution to give you this difference of 3 CQ values. You want your efficiency to be between 95% and a little bit over 100. These are the limits, 104, I think, or 105. Still, you have a reliable assay. The R square needs to be close to 1. And the Y equation that you see here in all these examples, the equation of your line is very, very important to have it handy for all your assays. Many people are asking me, what CQ value are you using as a cutoff? What CQ value do you use to call something positive or negative? CQ values, means nothing, right? Depending on the setting of the thermocycler and the bar that the software artificially puts somewhere to call something positive or negative. You have to have this equation handy. The most important thing is to, if possible, is to have this curve for every PCR plate that you run, but I know it's not practical, but at least for your assay, you should have this equation handy so you can translate the CQ value to copy numbers. And that gives you your answer for those marginable samples if the sample is truly positive or negative. If you have less than one copy of DNA molecule, you know that's not true, that CQ value doesn't represent existence of your microorganism. So very, very important to perform this validation step very, very carefully and in detail. Example of specificity, how we do it in our lab, and the importance of what you heard earlier for all of us to be able to exchange positive controls and have access to positive controls. This is an extensive list of true positives, either graft inoculated or tested by us or other labs are positive for the specific target. And the other set of controls in terms of specificity, we have true positives, you have true negatives, plants that have been through therapy and protected under screen. So the chances of being infected, in this case with Liberibacter, very low. And the other true negatives that we're using in our lab is non-pathogens. We're testing our assays against other graft transmissible pathogens that may be co-infecting a plant. Transferability, we collaborate, and that's the beauty of our network with other labs. There is different ways to achieve to perform a transferability validation step. Either you give your SOP and your sequences to the other lab and say, go ahead and run the reaction, or you send your materials and say, here it is, run blindly your samples and report back your results. In this case, we gave the assay to two different labs in our network. One, I think, and that's something you need to keep in mind, especially if you're working a lot with international entities. I think in this paper, we sent the assay also in labs outside the US. And robustness is my favorite. I like this test a lot. When we figure out a way to run it in our lab, we were very excited. And we knew, for example, that we could trust, in some cases, students or people with less experience on running PCR. So what this test did in our case, we intentionally pipette up to 2 microliters of the wrong volume of different reagents. Or someone went and messed up the thermocycler 2 degrees Celsius up or down. And of course, the CQ values will shift, but to my earlier comment that in our case, as long as you don't misclassify a sample as positive or negative, it doesn't matter that your CQ values went up, by 3 or whatever cycles. The important thing is that you don't misclassify a positive sample as negative or vice versa in the way we function. So we know that by running this validation test, that the assay, no matter the experience of person that runs it, or you drop something, and you went over to pick it up, and then you return on your plate, and you don't remember how much you pipette in a specific well, and so on, that the reaction will not end up with giving you wrong results. The newest tool when it comes to validation that I would like to introduce to you when it comes to validation of diagnostic assays or pathogen detection assays is the DAVN, or Diagnostic Assay Validation Network. This network was established with, or is described in a paper from 2003, is relatively new. We have not been able to use it yet on validating any of our assays. We're still experimenting and learning this pipeline. The pipeline is based on the MIKE guidelines. It's not something novel, but what this tool which is available in the American Phytopathological Society, you can see the very simple URL there on the top of the title. What it does is it's bringing all the tools that you need to execute the MIKE and to run your validation in one spot. Statistical tools, workflow, essentially digitalizing the checklist that you saw in the table. And the last two bullet points you see there under the tools is that you're having access to a community that does this all the time, and you can ask questions, and there is deposited resources and so on. Again, brand new tool, promising tool. In order to access it, you have to be an APS, American Phytopathological Society member. So as I said, we're in the process of using it and learning it. When I was checking the other day, the software online, I saw some familiar names and faces. So I know some of you in this call and definitely in NCPN are aware of it, and hopefully will become something very useful for all of us. I just wanted to introduce it to you in terms of resources. You have quantitative 2009 paper from Bastin. Bastin et al. Also have some very interesting supplementary publications on qPCR, so you should read for them. Then you have the 2014 paper from Borders for qualitative PCR, and now you have this resource available for you to experiment and play with. And as a conclusion, with my thanks for your attention, is that what we are one part of a systems approach to keep the specialty crops and the supply of food for our nation clean. We are at the very basic or top level, whatever you want to use, and what allows us to function properly is diagnostics and achieve our mission, but diagnostics that they follow in their production and validation, these guidelines and overall quality systems that we're discussing today. Because if we don't function properly, the whole system collapses. And of course, in the case of citrus, in this quick diagram, all the other elements of our systems approach to keep our citriculture healthy, at least in California, all of these steps are based on reliable pathogen detection assays. So what we do in our labs is very, very critical. Let's work together to keep the quality standards high and work together on applying these guidelines that we just discussed, so all of us can be successful. Thank you and for your attention, and back to you. I'm going to stop sharing.
Thank you. The next presentation will be on HTS by Maher.
Can you hear me?
Yes.
Okay, good. So let's share. Morning, everyone. So basically, I'll try to share with you some of the HTS guideline we are implementing here at FBS and also some other clean plant centers. I'll focus on the pipeline we have here. As you know, FBS is a multi-crop center. So we handle multiple crops from the one that handled in NCPN. And soon you will see in the slide here, we are adding berries other than strawberries, the other type of berries. So I think Georges did a great job with talking about the PCR as a specification and the MICA guidelines. And we are in the process of building similar guidelines for the high throughput sequencing. So if we want to say that, if we want to say how this will be done in a way that you have, it's a high throughput sequencing, multi-crop, it's a multi-steps process. So we want to make sure that we handle this in a very careful way. You have the showing now in the slide, the protocol that was approved by APHIS and the CDFA and some also other state regulatory agencies. But let's see what with that, you see that you handle the samples of multiple stages, multiple tests, and you have a combination of high throughput sequencing and PCR. I would like to share, if you are not aware of this paper, this is a great paper where we summarized the protocol. And as you can see from the title, it's a new era for the federal and quarantine certification and the testing that we do, and also the quarantine material. So this summarized the different steps, comparison between the old or conventional techniques and other techniques. Let's start with data collection. So when you talk about the requests, so you need to make sure that when we have a positive sample, the testing for that material, you will try to collect as much as possible information about this material. Let's start with, which is the time point. As I mentioned earlier, we test at the multiple time point, informative, time point 1, time point 2. And you will see that from that, what material you used, what's the date of the samples. And if you go in further details, that you will see that this is a positive sample, you will find a different requirement, while in the test that we run for the PCR are combined by also for the same plant ID, so you know that everything will match at one point. Database to track this progress and the process, you will see where while you are running informative high throughput sequencing, time point 1, time point 2, how many plants, who did that, what's the PCR result, and this is if it's ready to be released, everything is negative, that sample will be sharing the result with the APHIS and the CDFA to request release. If it's positive, this will mean that it goes to TCE, which is therapy, which we'll talk about later, to start the process. So what are the considerations when you're talking about samples? You know, virus titer can fluctuate between seasons, so we need to make sure that we are sampling in the right time of the year, right tissue, and also to address the titers. We want also to consider the virus distribution in the plants and make sure that your sample should be present all the quadrant of the plant. If it's a grape from both sides, if it's a tree around the tree. So try just to make sure that you are taking this in consideration just to increase your chances of detecting any low titer or unevenly distributed virus in the plant. We include internal positive control to evaluate success of the total RNA extraction, which we will talk about later. But also you need to make sure if you are testing for a pathogen, let's say you are testing for an Eller virus in late summer, we have to test for that sample, but you need to go out to the field and try to collect in a similar condition a plant material to include in your sample. So you have, if you get negative, for your positive control that you verified before, this means that your sample was not tested in the right timing. So you need to take in consideration to repeat this test so you don't get a false negative. And we do all the quality control for the RNA on the sample. Here are some examples of the different types of sampling material. We do both for grape petioles and we do park scraping because previous studies showed that there is a difference in the titer. And, you know, for different crops, sweet potatoes, strawberries, etc. So this is an important consideration that you need to take in your sampling process. And you can see here, as I mentioned, for the grape, we have to test it twice because we found some viruses can be missed if you test only with petioles. And so this is why we use different type of tissues. We use the black turtle bean seeds as an internal positive control, and that will help us for two things. First, help us with make sure the bioinformatic analysis was done correctly, and help us also with the process of library preparation, total nucleic acid extraction is done correctly. So with all this, we sample, put this in the bag. It's a very small amount of material, and with this, we will have the plant material. You have a virus, which is, we know that it's infected with some endonoviruses, and we use that one. We are all aware that if you mishandle the samples while you are processing them and sampling them, you will be dealing with possible cross-contamination. So how to avoid that? Gloves, gloves, gloves. Also, you need to make sure you see how we put the bag, outside, just to make sure that we handle the plant material. Also, bleach, whenever you want to work on a surface, you can see even we wipe the tables with those, antiviral and bleach and disinfectant. And some processing with high throughput sequencing, as we say, with power, great power come great responsibilities. HDS can detect a lot of background if you are not careful for cross contamination. So we put some plastic bags to cover. We use bleach and we use different ways to avoid cross contamination. As we say, you need to also to quantify your total nucleic acid after the process, especially if you are working with the new crops that you are, as I mentioned earlier, as a multi-crop center, high throughput sequencing can be used at different crops. So you need to make sure that you validate your protocols for total nucleic acid, and you make sure that also you quantify and validate your RNA or DNA. With that, it comes to the library preparation kit. We have a kit that we use here. We are evaluating different kits. We use Seq 500 as sequencer. We do all the steps as in-house, and we just upgraded to different automated liquid handlers. We upgraded from the Biomic to the Hamilton. That will be able to do 96 samples. And you can see from the process, we have to sequence multiple times. And we now doing everything in automation, and we quantify. And here is a good example of a good libraries of sequencing. As I mentioned earlier, we just upgraded to a NextSeq 2000. And this is basically, you can see from all this, as a multi-crop center, we do work with multiple. This is a big and a huge investment for us to have all those equipment in-house. So you don't need to start from scratch if you need some HDS sampling or testing. For example, we are working with Dr. Mark Fuchs, Cornell Grape Center, and also Oregon Department of Agriculture to test their material. And you can contact me or if you need any high throughput sequencing testing. And also we are working with Oregon State with some of the samples with our colleague at Walt and Christian. So the next step is to make sure that you follow our strict bioinformatic analysis. And in this case, I know that you will hear about the eProbe later, but here we are talking about the de novo, which is basically a method where you sequence for everything and you screen for everything. And this is just the pipeline. You have the mapping, direct mapping, or de novo sequence assembly, where you get a larger sequences fragments and you assemble. We just published a validation for our grape and what are the best practices for the analysis. And it's also with that, it's important to look for the next step. So bioinformatic analysis include curation. You need to look at the data. Whatever web-based or off-shelf that protocol that you use or pipeline that you use or open source, you need to make sure that you look into this data carefully and look at it from experience of, okay, if I see a grape virus in a strawberry, did this happen naturally, or this happened just for a cross contamination or a leak on the crosstalk during the sequencing process? So human expert biologists or expert technicians are extremely important to look at the data after you get the data for curation. So that will give you this is a real infection or a false infection. With that, so you need to talk about what's the host. You need to distinguish between what's the range of the host that include in the plant material. This is basically a real virus infection. This is If it's a questionable, you need to run further analysis. And if it's cross-contamination, this means that this is not a virus and you need to repeat. And that's, again, what you do here. When we talk about the curation, you need to confirm with an independent technique. So always, always, you need to go back, re-extract, test again with PCR or with whatever other technique, independent technique, to confirm your result. And just also echoing what Georges just mentioned, what we are doing here as a Clean Plant Center matters, and we are the original source of this, the major source of a clean plant material for different crops. So you need to know that whatever findings that you will be reporting, you need to make sure that it is verified. You need to use the right channels of communication with federal or state agencies, and make sure that you follow all the steps and check all the boxes to verify that what the work that you are doing is can be reproducible, can be repeated, and can be easily published with the finding that you have and can be taken in consideration. With that, thank you for your listening. I'm happy to take any question later.
Thank you, Maher. That was lovely. The next talk is still by FPS, and we're going to move on to therapy.
So next talk is therapy too.
Sorry.
I'm happy to. I'm happy
my
to
mistake.
talk.
Sorry. Yes. Sorry. The next talk is we're on diagnostics and on e-probes by Andres and Tyler. Apologies.
Can you hear me? Yes, I think so.
Hello, hello.
Let me share.
Yes. Hi.
Sure. Go ahead, Tyler. You can start because your second late's yours.
Okay, sounds good. Yeah, thank you, everybody. So today we're going to talk about HTS as well, but in this context of e-probes and kind of like implementation and guidelines, how and a little backstory of how this kind of came to be. Implementation of HTS for diagnostics. So with the decline in costs and significant improvements in HTS, it's really allowed this to be used for more routine diagnostics. You can set up these instruments relatively cheaply in-house, with example with the nanopore sequencer, or you can get these outsourced to different companies or facilities out for very affordable pricing. However, one of the continuing challenges of HTS has always been the computing infrastructures. You're dealing with gigs and gigs of data, which require specialized workstations, or in some cases, AWS clouds, which is not the most trivial thing to set up, as well as the requirements for bioinformatics expertise. This is where we feel eProbe diagnostic nucleic acid analysis platform, or ETNA, it kind of bridges that gap, where it's a really user interface friendly system to help analyze these data sets in silico, and it helps with a lot of reproducibility issues that were talked about in the previous slides or previous talks.
So with Edna, what we use are eProbes, which are unique signatures, genetic signatures, which is a little bit different from the traditional analysis of high throughput sequencing, where we map to reference or we do blast, as we saw in the previous talk. So in this case, what we do is we select those unique regions for each of the pathogen, and then we have called them eProbes, and we have a program specific for doing that. And it's a graphical user interface. And we can design EPROBs depending on the target of interest. For example, we can do it for genus, we can do it for species, or we can do it even for strain. And that depends on what's our final objective. Most of the time we do it for the species level. And we always try to, every time we develop an EPROB, we want to go through different steps of validation. It's going to differ probably a little bit from the traditional analysis of validation, because we have the step one, which is EPROB design, and we published in 2021 what would be the process for evaluating and validating EPROBs for HTS detection. And you can see here 5 steps of validation of EPROBs. And with step one, which is EPROB design, we need to have as many target genomes as we can retrieve from public or private databases, and also as many near neighbor genomes as available. And our system, which is called Microbe Finder, the user interface is called Microbe Finder, where users can create accounts and do all this process. Our system, what does is it generates these whole genome comparisons and selects automatically those EPROBs, which are only curated to the level of your near neighbor. So your EPROBs are going to be as specific as how close your near neighbor is. And also we don't have our host genome here in this comparison. So what we have after these comparisons are raw EPROBs. And then we go to step 2, where we further curate these EPROBs, and we use BLAST in this case, and we often use the NCPN, and we have scripts for eliminating EPROBs that could be cross-reacting with other organisms, and also with the host genome. So we tend to use NCPN and the host genome as the first tier of curation. And if, let's say, we have an EPROB data set of 100 sequences, that could reduce the number. And with reducing the number, we also have lower sensitivity. So we have to find a good balance between sensitivity and how much curation we have. So we have this part of the curation, we're going to have false positive EPROBs. Those are the ones that are eliminated. And then we have the curated EPROBs, which can go to step 3, which is further validation of the EPROBs. And the validation of the EPROBs within step 3, what we do is we do in silico validation first. And initially we were using high throughput sequencing simulators, trying to mimic serial dilutions of the pathogen in a host, and including the microbiome that's usually found in the host. And we were using originally, as I mentioned, open source software that were available, but many of them did not have enough support. So we came up recently with a new simulator, which helps us do these validations. So we use MESTank, which is specifically designed to do these serial dilutions. The goal is to find the limit of detection and analytical sensitivity in. Can you hear me? I think my.
Yes, we can.
Oh, sorry,
Yes, we can.
Sorry, something is going on with my camera, but let me turn it back on. Let's see. Okay. So and find analytical sensitivity. And after that, we go to in vitro validation. And in vitro validation, in this in vitro validation, what we do is ideally we have serial dilutions of the pathogen in host background. So this depends on the availability of how much we can do the dilutions. For viruses, it's a little bit more difficult because you cannot culture them, so it's more difficult to do the serial dilution. So this was a proposal of in vitro validation for step 4, but ideally we should follow similar guidelines as a qPCR, I would say. And then we go to step 5, where we go to field collected samples, and that's where we've been developing with the CCPP limit of detection using in vitro collected, and also positive controls that they have in their greenhouses. So this could be greenhouse samples or positive controls in trees for the final validation of EPROC. And then after those EPROCs have been validated, what we do is we put them in a system that it's only for detection.
Yeah, so once we get these validated eProbes, then it's ready to be used directly on samples. So for citrus specifically, we use stem samples. They go through nucleic acid extraction, library prep, and then through sequencing on either the Illumina or the Oxford Nanopore platform, depending on what the needs are. And they go through analysis. Specifically with the Etna, you take your data that gets generated from Illumina or Oxford, you upload it into the MyFi Finder platform, where you can just pick and choose exactly which pathogens you want to search for, and it will automatically in the background do the statistical calculations and determine whether that sample is present or absent for the pathogen. As good practice, it's also good to store this data long-term somewhere safely, whether it's on a local drive or an S3, because that data is very valuable and can be used for reanalysis. Next slide. So during our initial phases, as a proof of concept, we designed 4 eProbes to target specifically citrus tristeza virus, Exocortes viroid, Liberibacter asiaticus, and Spiroplasma citri. Again, during the initial proof of concepts, these eProbes went through a very stringent curation process that Andreas highlighted earlier, where it be mapped against, where it would be designed against near neighbors, mapped against blast against the genome through any non-specific eProbes. And it also went through a pretty rigorous statistical limit of detection test. After the rigorous validation process, the assay was further expanded from 4 to now we have 20 fully validated citrus pathogen eProbes. As part of the validation process, we developed a new statistical model to help us determine these limit of detections. This statistical model is called the quaternary discriminant analysis, which basically what it does is that the QDA estimates the limit of detection. We feed this model with samples that are true negative as well as true positive. It's listed on the right side in the bar graph in the docs plot. From that, it's able to from the model. After feeding that data, we're able to determine whether the limit of detection is any given sample based on the total score. And on the right of the graph, you can see, or right of the table, you can see for any given eProbe, you'll need a certain amount of a certain score to determine whether the sample, any particular sample, is a positive or negative.
And after this final validation with positives and negative samples, which come from the positive and negative trees at CCPP, if we end up with a good limit of detection, we move the eProbes that are ready to be used to a platform that it's only for detection. where users can select the EPROBs that are validated. So these are EPROBs that are fully validated, and users can submit jobs without having to deal with the other user interface where users are only designing EPROBs and validating EPROBs. So we have two platforms. We have one platform for EPROB design, and then we have a platform that it's only for detection, where we only include EPROBs that have gone through those rigorous validation that we mentioned with Tyler. And this is an example with one Citrus one Citrus HTS run. And in this, we included, I think, all the as many EPROBs as possible. And this was part of the evaluation of the EPROBs using this system. You can upload the metagenome here, and then you run it through the test. And then at the end, it's going to tell you with a p-value if it's negative or if it's positive. Whenever you have a p-value equal or lower than 0.05, it will be positive. And usually having a very, very low p-value might give you also a hint of how concentrated your pathogen is in the sample. And that's the end goal. So users can have a easy to use user interface for doing detection using high throughput sequencing.
Yeah, we feel that Aetna addresses a lot of the major concerns with HTS, mostly because it's a very user-friendly interface, and it spits out a nice report for you at the end. It's really straightforward that basically anybody could use. It's a scalable platform. There's no major commitments in terms of infrastructure and computing power. It's scalable. It can be implemented at various centers. The good thing too is also, you know, you can continuously update and design new eProbes as pathogens emerge and further characterized. Right now, for citrus, there's 20. We can detect up to 20 different pathogens. And I think I mentioned this before, but any old HTS data can be quickly reanalyzed for any new pathogens that emerge without needing to re-extract or re-assaying anything, further making this a lot more efficient in the long term. And that's it. That concludes our talk.
Thank you. Thank you very much. And now is the therapy talk. Thank you. And we'll be taking questions later at the end. We can take questions during the Q&A session. Maha, can you hear us?
Can you guys hear me?
Perfect. Thank you.
So we talked earlier about how to detect the viruses. You know, PCR, real-time, or eProbe, or high-throughput sequencing using de novo. Now, the next step, you know, NCPN goes with therapeutic and diagnostic. When we talk about therapeutics, this includes trying to eliminate the virus and verify that you eliminated this virus by multiple process of additional testing. So when we say cleaned here with that plant material, means that it's tested or free of economically significant or regulated viruses or pathogens. We talk here about multiple crops as a multi-crop center again, grapefruit trees, strawberries, and sweet potato. And the idea that you want to do the meristem tip culture simply, and this is an example of Nicotiana pentamiana shoot where you can see the virus that in the protoplum, you want to cut the meristem before this virus reaches to the meristem tip culture. So you start with collecting the plant material, you do excision, you transfer, acclimatize, advance to container, and then testing again. So you make sure that you were successful in eliminating the virus. Now, let's say before the approval of the high throughput sequencing with combination of PCR protocol that I shared with you earlier, what was the common practices here at FPS? We have to send, we will do like a presumptive we will do a presumptive HTS, not HTS, sorry. We will do presumptive virus elimination. So we will send the plant material directly to the tissue culture lab to start with the process of virus elimination before we conduct the biological testing and finalize that. Why? Because before we don't have we cannot use high throughput sequencing because it wasn't approved or developed at that time. So you need to go and test both the source material and test the material that you run tissue culture and therapy on. This to make sure that you are not waiting 2 years for the biological indexing to finish and then start the testing of the tissue culture plant material. So we were doing this kind of in parallel when we get the plant material. So that's what we will call the presumptive virus elimination or therapy. Now with HTS and the informative testing that we conduct, which is the first test, high throughput sequencing test we conduct in the source of plant material. If the plant material is infected, we'll send it directly to the tissue culture lab. This saves money and saves time for both us as a clean plant center and also for the customer. And we are not wasting resources and give an informative result faster to the customer. So with most of what we receive, yes, is positive, but if the plant is negative, there is no need to run a presumptive therapy on it. So from the slides before, you notice that this is a long process. Takes years from do the virus excision to do the plant excision and to remove the plant material to the point that you have a final product that you want to test. So we implement the buddy system here. So 2 people will make the collection and verify the record. The QR codes are used for when we collect the sample, and we take a photo of both the plant material and the plant and the QR code and the label that we collect the plant material. May you think that this is basically too much work, but if you visit one of our fields, let's say the grape, especially, you will see that plant material, especially from neighboring vines, can interconnect together. So we don't want to get to a situation that you are collecting one shoot from the neighboring vine, and it will be easily mishandled in the later steps. So this is save us time overall, even if you feel like there's 2 people doing it. When it comes to the tissue culture lab workflow, when we transfer the plant material, every 2 weeks or a month, you need to transfer the plant material. We work only with 1 lot, and QR code now are in all the trays. You can see the old labeling system, and you can see from in the right hand the new QR code system that we implemented. This is the physical labeling of the plant material. This will reduce the handwriting, which reduce error. And this comes hand in hand with the request of the plant material after the testing, which is what we have presented before. When the plant material is tested and it's positive, directly we will request a tissue culture excision for this group, and this will move forward. So it will come to the tissue culture, record keeping here. And if you look down in the slide, you see that this material was fresh material collected from Screen House 2 by two people, Vincent and Marcus, and the plant material, all the steps that you handle the plant material from collecting to transfers with a very detailed transfer records, and all this done in one place. This will help us to avoid mistake and increase the traceability of the process. Then we move to the acclimatize. You move to the soil, acclimatize it for 2 weeks on the growth chamber, and then you have it in the open in the shade house or in the greenhouse. Like what I mentioned earlier, that we want to sample, we want to do excision in this best case scenario here, that you were successful. We are good and successful within one overwintering season of testing, but because of the process, the virus titer can be very low. We implement, especially for grape and fruit trees, 2 overwintering season to run the test, the final test before we release the plant material. And this will give us the chance for any low titer virus to reach to a detectable level. So we are basically verifying that the virus elimination process was successful. And in case of grape, therapy success is more than 95%. We continue to learn and improve. For the multi-crop center, we have now more success with the fruit trees. And we just recently, thanks to the collaboration with Guy Lavok from Colorado, we are now implementing the cryotherapy for helping us to eliminate the viruses in fruit trees specifically, or viroids in fruit trees. And we continue to improve the process of virus elimination. So we just hired a consultant for 1 year. This was not done with NCPN money, that was FPS money. And we are improving and expanding our tissue culture lab based on the success. We continue to resources and testing and space is limited. So we want to invest the time to detect any positive plant material, means failed virus elimination, as early as possible. So we are trying now to test the plant material in the tubes. If it's positive, we will remove it and eliminate it. If it's negative, we will continue testing it, because negative result here you cannot trust. But if you are careful and you get a positive result, means that the virus elimination was not successful. So you don't need to occupy the space in the growth chambers or in the greenhouse with that material. You start over as soon as possible. So as you see, it's a system with a different multi-steps. It goes hand in hand with the diagnostic, but record keeping, cutting the excision of the right tissue is very important, and following the protocols for different crops and the variation of the timing is very important to consider. With that, well, thank you, and happy to take questions later.
Thank you, Maher. We can now move on to the final part of this section about maintenance and distribution, with the first talk from Imana Power.
Hi, everybody. I have this issue with my camera. Adjusting it. Sharing my screen now. Thank you for this opportunity. Hopefully you see the screen, right? The presentation screen. Only.
Yes, we can see it.
Okay, perfect. So yes, I'm Imana Power, LSU Ag Center, and I will present what we do at maintenance and distribution, mainly from the LSU Ag Center point of view, but it's on NCPN sweet potato. So, and sweet potato was added to NCPN around 2015, and we consist of 6 centers in total: University of Arkansas, Hawaii, NC State, FBS, of course, Mississippi State, and us, LSU X Center. These are the pages if you go through NCPN website, the National Clean Plan Network. All centers are capable of are receiving breeding lines and have heirloom plants and are capable of therapy and testing. And some do maintain tissue cultures themselves and do greenhouse increase either at their location or in collaboration with another institute. So this is a beautiful overview created by Dr. Clark that represents what we all do. I will focus on Louisiana State just to give you a few examples, but we follow similar processes as was clear to you earlier. So this is mentioned just now. We do forest elimination using meristem tip culture that can take several months. It starts with receiving the roots and takes several months. So we get plantlets that we take to the greenhouse and then use biological indicators with graph testing on biological indicator to test for to see whether or not our meristemming was successful, and then we continue with with the molecular test for iris testing. We still use this system. We hope that we can upgrade to HTS, of course, but that will mean quite some upgrading. Now, the main part, what I was will focus on is maintenance. Most of the centers that maintain their tissue culture plans have several different sets of tissue culture plans. In our case, at Louisiana State, we have over 200 lines that consist of breeding lines from the sweet potato breeders, but we also have a few copies of breeding lines of other universities for some testing, collaborative testing that is done, as well as some lines from SIP that we then keep in our tissue culture lab. Our system consists of these racks with lighting, all at a specific temperature that is tested. There's a light system, light dark system that is specific to sweet potato. And we have both a working collection and a long-term collection. The working collection, work collection that is up is every 2 months. We redo it, whereas the long-term collection we have for 6 months, and at some point there was talk to upgrade it to up to a year. Once we have our plants cleaned up and we know that the specific lines are requested by our customers, we then start our micropropagation to be able to distribute plants to for greenhouse increase. In the past, we did a lot of the micropropagation in the sweet potato lab ourselves here in Baton Rouge, but now we also work together. We outsource part of this to a specific company that you could see on the slide. We deliver up to 1,000, some thousand plants to our to Chase, where the increase starts in the greenhouse. And this is the Sweet Potato Research Center that is 75 years old, a little bit older than that. That is a combination of NCPN and the foundation itself, Sweet Potato Foundation. Their propagation starts with our tissue culture plants that we micropropagated. Either they receive it from us and then propagate it in the greenhouse, and that takes several months as well. This process is all documented, and this is where NCPN stops, and then the foundation, LSU Foundation, takes over to produce roots and seed bins that get distributed depending on the request. All of this is documented in our QMS that we had, that was from 2020 was developed. And we are still, as you can see here, by Dr. Clark and Kathy Robertis and Elisa Tubana did a lot of work. We're updating this now to make sure that we do what we say. And once that is done, and we've shared all of these documents with the different sweet potato centers, but once it's all updated, we should share that in the Google Drive as well. Just to give you an example, so we have several documents, forms, records, quality procedures that is all based, it's all documented, it's all recorded in Teams, and that is updated and as needed. And every week, once a week, this gets stored and recorded by LSU itself. Just a quick overview of what our work instructions look like and the forms that we have for either tissue culture preparation, long-term media, and how we track our tissue culture plans. Because we use a barcoding system, the old barcoding system, and we're upgrading it to QR codes. And we want to include all the information that we have from the first cutting all the way till the virus testing in there as well. Basically ending with this again, to give you an overview of the 6 centers, what we do, and where do we then, that all of us basically distribute our lines to specific customers wherever they are. It's requested. That is it. If you have specific questions, I'm okay.
Thank you, Imana. So the next talk is going to be a recorded presentation from Scott Harper, who sends his apologies for, but he couldn't attend. So let me see. So I'm going to share. You should be seeing that. And once I hit the start, I'll be very quiet so you can hear the presentation. We're going to try.
Everybody, I've been asked to talk about the impact of quality management on I'm going to use our HOP program as an example, on maintenance and distribution. What do we do? How have we learned? How has this impacted our program? So really, when we started our QM system, we really had to identify first what could go wrong. You know, if we think about maintenance, we're thinking about our foundation. What are the risks? Where are the problems going to come from? You know, what could possibly go wrong? We could get the plant IDs wrong. Problems with the disease status. Is there risk of introduction and spread into our foundation? Where's it going to come from? Is it planned? Are there insects? You know, how do we identify where these problems are going to come from? And just really be honest about it. What could go wrong? And not think, oh, that'll never happen, because that's usually the thing that's going to happen sooner or later. You know, what about the state of the facilities? How's the state of this infrastructure? The state of repair of everything? All these kind of things you have to think about when you start to build a system to make sure that your QM system really is impactful and makes it work for you. And then you think about it downstream from there. What about your orders, your shipments? You're sending out plant material to people. Is it the right material? Is it infected material? Hopefully not. But, you know, what do you do about that if that happens? So all of these factors play into building a QM system that works for you and really can impact the program. So if we just sort of think about our maintenance of our foundation, so what do we do to monitor and mitigate practices such that we don't have problems in our programs? And so, like, for example, we have potentially vector transmission of pathogens, okay? So we decide to keep the plants in the greenhouse, we have spray crabbing, and insect traps are placed and inspected. You know, if they're holding screens, we have monthly log inspection of the structures, soil-borne stuff, get it up off the ground. Make sure that we don't have these things coming into the program. At transmission, you know, we send SOPs in place around all these things so that everybody knows that the system works, things are checked, things are done properly, and these problems don't happen. And it's really all we had to think about is what are the problems and how do we deal with them, and put something in place to deal with them. Because the QM is not just about having an SOP, it's about actually following it. It's about making sure the infrastructure, all the pieces are in place, and then making sure that everyone's following the instructions. And really this comes down, as we've learned over time, document everything. Without it, you can't get to the root cause of a problem. For example, this is our inspection form. Every month, the staff go out, and with every individual facility we have, they carry out their inspection. Series of questions and things to look at that may or may not be a problem. Additional notes, same thing for the plants that are in those facilities. They look at the plants, they record everything. And then if there are any problems, it's identified, reported, and can be followed up upon. And that's really what this comes down to for a QM system that works, and something we found is necessary to have a functioning system, is to be able to identify non-conformances and make sure we have documented corrective actions. This is necessary for improvement. So we have a non-conformance report, all the details go in there, which largely leads to corrective action requests. So, how do we fix this problem? What do we need to do? All the steps that are laid out. And then finally, all this feeds into continuous improvement. How do we make sure that this thing doesn't happen again? And this is really what it comes down to, folks. Making sure that your system works and that you get better with every month, every year, you're iterating and becoming better and better. It's not an overnight process, it's iterative. And in the 5 years since we started implementing our QM systems, we've seen, particularly in the Hop Foundation, but really across all the crop systems that we support, reductions or eliminations of no breaches or introductions of pathogens into our foundations. Things aren't coming in, which is good. We're identifying or removing anything that doesn't meet modern standards, making sure that no incorrect orders are being sent out, because everything from plant to packing, shipment details, everything is checked, rechecked, and recorded, so that if something goes wrong, we know what happened and can fix it. Because no one likes a customer appearing and starting to yell at you. And really what this has all come down to is, by fine-tuning this process, we've been able to reduce our overheads, both in terms of labor, time spent on fixing things, and just really focusing on refining and improving our systems. So yeah, it's not something that happens overnight, but adopting QM practices and implementing them in your program can have significant impact. Thanks.
Okay, thank you. All right. So we were supposed to have another talk on distribution from Texas, but we got a message with apologies from Mark Van Ness, and he was not sure whether he was going to be able to join us, and I guess I don't see his name unless I'm missing it. And so I think I want to thank all of our speakers, and I think we're ready to move on to the next section of our workshop, which is the Q&A. So let me pull that up. Okay, and share it. Oh, so please do go ahead and check the chat, because there's a couple of people we don't recognize the names and we don't find the registration. Or maybe they've joined using someone else's Zoom account and is showing someone else's name. Just please go ahead and check and let us know who you are. And let me share in the chat the link to the meeting chat. Okay. Everyone in the meeting. Okay. And you should be getting, please let me know if you can access the Google form for review, feedback. In the meantime, we're going to pull it up and share it, if I can find it, with my documents. I don't want, no, it's not letting me do that. Okay, here. All right, can you see the Google form?
Yes.
Okay, thank you. And so
Irene, let's confirm that everybody is able to access the link. If anybody can tell us, yes, they can. Okay, we got one confirmation. Okay, let's go.
So on, yeah. And so this is the time where if you want to turn your cameras on and unmute yourselves, and we can have a conversation. We can open up the floor for a conversation. This is not just the forum, and we thought we'll put it up on the screen. If you want to leave your name and your email, but it's optional. If you wanted to fill it out in an anonymous way, we're equally happy to get the feedback. The questions are, what is one actionable takeaway you gain from this training? Do you feel confident in applying the quality management concepts we discussed today? Why or why not? What specific areas or challenges at your center would you like future trainings to address in more detail? And the last question is, if your lab center specializes in a particular procedure, would you be willing to organize a workshop? If yes, please specify the topic of technique. And so we have a few minutes, and I think all the questions are very important, but we would like to focus on, given the fact that we are on a tight schedule, on questions number 3 and 4. However, if you have thoughts about also the other questions, please go ahead. We're more than happy to listen. The idea is to stimulate the discussion. That's how we devised the agenda, and we hope you enjoyed it.
I have a general question, Irene, for everybody in the system, in the workshop. If centers can share if they have a dedicated quality manager position, or the director executes, or everybody participates. How is the quality management structure in terms of a specialized dedicated position or a different model?
I can speak for Luis for LSU, Sweet Potato. We're actually a research lab, and we do service too. So we don't have a dedication to this yet, but of course we're open to expanding and upgrading. So it's basically now still 3 of us, Kathy, who's the manager, and then Kathy DiRoberta, she's on, Eliza Tubanya, and myself. And now we're trying to include people. But we work on sweet potato post-harvest diseases as well. So it's not just viruses. All of this, limitations are usually funding, and that is linked to facilities. We were grateful that the facilities in our lab are upgraded due to NCPN funding. And then it's human resources, of course, not just the number of people, but also the skills, the skill set to devote to this and upgrade.
Thank you. Yeah.
Yes.
So you started your statement that you are a research lab, right?
Yes. Yeah.
And
Yes.
and why did you put that up front? That that quality management came up only when your lab start getting into the clean or pathogen tested material. It wasn't an issue before you picked up the NCPN tasks.
Right. So this position, I started this position in September 22, but Dr. Clark, who had this position for 45 years. This is basically a faculty position. And with his research, he saw the importance of viruses in over the years, of course, he saw the importance of viruses in sweet potato and did a lot of work to get, sweet potato added to NCPN. Because a lot of the production is, yes, vegetatively, right? So we have the accumulation of viruses. So since 2010, I guess, all these viruses, the polyviruses, chlorotic stunt and leaf curl have been identified as yield limiting viruses. And now we're of course working on identifying more with this multi-state SCRI, Specialty Crop Research Initiative, Clean Seed, to see if there are more viruses that we should pay attention to. But it's basically created, I don't want to say created, but the attention to viruses being this important in sweet potato was through all the work, collaboratively, of course, but led by Dr. Clark. And so it's a faculty position that has NCPN as well as surface. There are specific centers that are dedicated to clean plant, right? It's really viruses. Some have more crops, like FBS, NC State have different crops, but that helps with increasing the facilities and human resources. Did I answer that correctly?
No, there is no correct or not, but you gave me the background for your answer. That before all this accumulated knowledge, not anybody, there was not much emphasis on clean stock, but then clearly there was research identifying viruses as a limiting factor. And then as the center was established and kept evolving, quality also became part of the equation, right?
Yes, but let me clarify. So
Yes.
they worked on viruses, and there was tissue culture work done, in the early 2000. But identifying that these 6 were yield limiting was around, and that we could detect them was around 2010. Yes. Yeah.
Okay.
But so that's how long it basically took to identify. Hey, we do have these are viruses that are causing these symptoms and are yield limiting, and we should be able to identify them and clean up sweet potato.
Okay. Yeah, my comment was more general, and I wanted to use that initial statement with a research lab. We were a research lab, but not specifically for your answer, but my general comment would be that this quality management system should be applied
Throughout.
through and through. Yeah.
It
never crossed my mind that we need such a system for our research before the training, and we need to acknowledge the NCPN that opened this door for us in terms of plant systems and plant centers using these principles. NCPN introduced all of us to this concept with the early trainings in Iowa State, with the veterinarian labs and the USDA facility there, the high-level facility, work with anthrax and so on. So we're ever grateful that we were introduced to this concept and for the support of the NCPN to continue beating the drum of quality. So I'm now preaching to the choir, but for all of us on this call, we need to spread the word that no matter what you're doing in your lab, if you're going to put, I made a comment, Maher repeated it, and I'm going to restate it. When work is published from our labs has implications. It's not just another paper that describes a gene or a plant or a pathway, and a researcher may or may not pick it up. The work from our labs have implications in real life, in regulations, in laws, in businesses, in trade, in exports, in imports. So quality should dominate our lives and our works and our work through and through. That's That would be my comment, Iman, and thank you for your answer. Yeah.
I can add that we are basically translating this, yes, to our other research projects. We're working on sport trapping in a specific company. And yes, we're basically having all those protocols that we just used to have. We are adjusting them to the QMS as well. Yeah, that's true. We apply it to everything. I'll just quickly answer while I'm talking the question, what is the new QR code platform that we're updating? So we have on our tubes, I should have taken that picture better. On our tubes, we had the regular barcoding, the name of a breeding line or a cultivar that we have in tissue culture. And we would have the name and the old barcoding system, and now we're updating that to QR codes. But we should also link that to our information system as to when we did which testings and if we ever repeated the testings or not, the nucleic acid extractions we did. So all of that, the meristemming, we have those, we need to link those documents that if you are with your barcode scanner in a tissue culture lab and you scan, you should have all the information from the time you received the root or the time you received the tissue culture plan from a different lab. And so you need to just connect the two.
Imana, there is one more for you. What information system do you use?
So it's I love how you say system. It's you need the brother system, the actual where we print our codes on, because we don't have limbs. We don't have any of that. So we really use all our documentation. What you mean, the QR scanner, Kathy? Do you remember if you're still on?
We actually, yeah, let me say.
Go ahead.
We actually use, I was just typing that into the questions and things because I keep really hoping we have somebody who comes up with a great limb system just for us. But we use an old, I think it's 2007 FileMaker. And we've created our own file where we manually input all this information. And like Imana says, being able to scan something in and have everything connected would be ideal. But over in that angle, we have the FileMaker system. And in the other angle, it's just an Excel spreadsheet that's attached to our barcode scanner. And that's just saying the name, the data was transferred and things like that. So unfortunately, they're not linked. And I don't know.
Not yet.
Yeah, right. They're not linked. So
working on it. Yeah.
Yeah.
Let me see if I can be quick.
Yeah, this is what you are discussing, Kathy and Imana, is natural evolution of things. Even in time flies. I've been with the program 25 years. 20 as the director. I came as a student in the program, and it was all, when I first came as a student, it was all on paper. Excels were used a little bit. My professor all day long for years, all he did was updating an Access database. Looking at paper and typing, looking at paper and typing. The Excels were used to design maps. I found it very interesting. The Excel spreadsheets were not used for entry points and analysis and sorting because it had nice boxes. The cells on the table, they were using them to design field maps. Because these were, as I say, and I could see it in Word documents as, computers and software was coming into the program. And the typewriters were still there. They were using the software as a typewriter, right? They wouldn't align things in the center by aligning in the center. They will hit tap, 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 tap to bring the title in the center of the document, right? Different generation. And so when I started, I realized there is no point of keeping notes in one thing and then transfer them. And it has to be a live system. To Kathy, to come to your point, what system we are using. We have tried off the shelf. We have tried the Microsoft Office available things, Excels and Access, and we have tried off the shelf LIMS. At the end, nothing worked, and I think Maher can verify from our conversations with Deborah Golino early on, and then Maher coming in. What really brought everything together, what you're describing, is in-house IT support. A programmer in the program, inside the program embedded, either a contract person, whatever you can negotiate with your institution for 2, 3, 4, 5 years, or permanent position. So the programmer lives with the program. They actually physically see your pots and your tubes and the greenhouses and the fields and the people working, and they translate in their head with our help. They translate in their head what happens in the real world to code. And that's when everything fell in one spot, in one place with us. So nothing happens right now in our program, and you saw it also in Maher's screenshots, outside the system, live system in the cloud. Yes, there is Sharpies and pencils and papers, but it's like 0.00. You make a quick note, but in reality, you're scanning, you're typing, you're using your phone, you're using your laptop. We have these little tripods that there is laptops all over. You can put your laptop everywhere in the greenhouse or in the field. And unless if you get into a live system that everything is recorded real time, you will always have this disconnectivity issues. And my last point on Excels and paper and so on, all our paper records, to a great extent, we need to find another student to continue the project. They have been scanned. We have been scanning all our paper documents, and all the Excels, the important ones, have been converted to Google Docs. So the scanned documents are attached to the digital entry into the system, and the Google Docs have become links and shared documents inside the system. So you can jump back and forward and in and out. And so these are my few thoughts and my experience in terms of having one system monitoring all your process to be able to maintain quality.
Totally agree. Thank you, Georges. Very true, all what he just mentioned. And it's very important to highlight the last point that Georges mentioned is going back to the old records and digitize them, and everything will be scanned. Ordered for 60, 70 years old, order in paper or cards now is scanned. Maybe you think has no value, but in in specialty crops, everything has a value to be traced and to know where the things comes from, and it's important. So I agree. Thank you, Georges, again for very nicely summarize the current situation.
Yeah. So you can try to buy all the software and play and so on, but for me, talk to your administrators, whoever supports and understand the idea, and just go straight to what older programs like FPS and the CCPP we went through, because now the technology is also there, and just go straight to create your software. And then when you get, if you manage to get your IT person, we will bring you in contact. We will connect you with the CCPP IT, and Maher, I think you have your own. We will bring them in contact. They can even take code or they can brainstorm quickly because the pipeline is the same.
Okay, careful. If you promise
Yeah.
me something, I will not forget. I will remember. And
The stars
And
there. Yeah.
we tried early on, and I don't know how many in this call know that probably Maher was around. There was a grant, NCPN, many decades ago, to hire one IT person to build a system for everybody, the whole network. but it didn't work out. It was plus again the technology was not there and the structures was not there. But yeah, your IT, no reason for your, if you manage to get someone, no reason to start from scratch.
Right, exactly.
Yeah.
I can quickly, if I can share just briefly in 30 seconds what our system looks like and that we do. Can I go ahead and stop your sharing? It was really quick.
Yeah. Go ahead. I think you should be able to kick me off, and then as you prepare. So what I'm hearing, I'm finding it very interesting. What I'm hearing is we are talking about those priorities about data management. Really, if we're talking about the records, even the old records, and I think to echo also what is coming from Georgios and Maher, this is what we're really seeing in the centers, that it's about the integration also of the different systems, because all of us or most of us got involved with the barcoding, and they have something or they have something else, and it's getting these systems to talk to one another or in a format that is helpful to you and it's functional. And I think that the dream of the LIMS, that once you customize it, then it does everything. It might be out of the scope of what can be afforded or what is actually even functional, because it's not so easy to customize these limbs. So I think in the, well, you're on the committee, you're on the steering committee for quality, Mana, and that one of the things in the grant and we discussed that we wanted to talk about, and one of the trainings for the future was giving an overview of but digging deeper into the sort of limbs or like limbs that people use in order to manage their data with a little bit more detail. That was one of the ideas of the training, and if that's what is emerging from this call, that is what people want. They need more support in terms of data management. They want to know more about that. That is certainly something that we can do. I do want to point out also that so far we have received 3 responses. I don't know if people, they're enjoying talking and listening and they want to do it later, but please do submit those forms or talk to us. Yeah, open your mic and talk and share your ideas, because that is really important in terms of what is going to be offered in the future. Okay.
I agree. This is just a quick overview. This is the file. I think you can see it, right? This is FileMaker, and if we then want to have an overview, overview of what it looks like, this is then what we get, which works. And this is what we can share with others. But it's really this. I keep clicking the wrong one. It's really this big overview of data, and it's with a million columns is what Kathy tells me every time. And so not all of this has it here, but the information is recorded. All of it first on let me see if I can share the other document that I have open. First on this is just qPCR sharing, that multiplex PCR that we share for the PODI that we get a hard copy document that we print and then we scan and upload it, right? So it's a lot of work. You either write it and then you scan and then you upload, or you write it in a document in the greenhouse and then you type it in and then you have to upload it. So, I mean, I say a lot of work, but this is Kathy. She's she's doing all of this, and it's a lot of work. So anything we can, I know the breeders have a field-based app that they're trying to push, and anything that we can develop that can, like Irene said, that just is not too cumbersome, that works for the system you have, and that connects all the collected data already. That is what we would love to have, and we're working towards, And it needs to not only work for us, for Kathy, Liza, Imana, but it needs to be consistent for all NCPN SP and then everybody that follows me. I'll stop sharing. But thank you.
Yeah, this is all normal what you're describing, Imana. It's part of the evolution. But networking, that's why we're here, to exchange ideas.
I will definitely email you. I
Yeah,
remember where you talked about, this is another refresher training, where you talked about the audits, the internal audits, and it's on my to-do list for a while. That is also part of QMS, but
Yes, we call the walkthrough. Some of you are referring to me, Mana. Yeah, the walk. We bring them in. Yeah, and by no means. And again, please, as Irene said, unmute and share your experiences. By no means don't think that we do everything perfectly, right? We forget things, we drop the ball, we make mistakes, we have the what the hell happened moments. This is all part of the process.
Yeah.
But definitely we are in a completely different level from what, again, the program used to be when I started. And whenever something goes wrong, very quickly you can trace it because everything is in place. And I think from you, Iman, again, when you talk about not only the resources and the facility and the people as available FTEs, if I can call it that, but the quality of people, right? And what their background knowledge is, and what's the way they approach their work, especially for clean plant centers, very important. So Irene, I would add on your list what's happening today. It needs to keep happening.
Yes.
And not only for the leaders of the programs. I don't know if you can have these kinds of trainings, refreshers, whatever you want to call them, all the way down, like to the person that cleans the floors. It doesn't matter what they do in our programs. Our programs require a specific set of mind for the program to be successful. How people are approaching the jobs, as I say, cannot be 9 to 5. Your tasks needs to absolutely consume you. You have to live this idea that you have to follow the protocol and you cannot cut corners, and you have to deliver this product. Otherwise, there is real life consequences. So this constant training and reminding for all of us and all the members of our teams, I think, is very, very important.
Thank you. Yeah, this is one of the things that also emerged from the think tank. There was a comment that we heard several times back in June that it is important even for the members that even in the steering committee to say to have these events, and the fact that they don't take too long. There are these workshops of a couple of hours, 3 hours max, something like that. But frequently they get things into motion, and someone may use a certain type of language and use some keywords that just reminds you of a concept and keeps it fresh, and gives you an opportunity to network. I think one of the recognized, the highest probably recognized value of the quality initiative is that it's an initiative that really brings together all the different commodities, and we're very proud of everybody in the network for supporting this and participating. And that's something that we want to continue offering. And so we just we don't want to offer a workshop just because it was written in the grant, but it doesn't reflect the current feeling, the current need. And that's why we constantly ask for feedback to keep that finger on the pulse of what the community needs, what's going to be most useful now. As you know, we submit a grant in August of a previous year. We don't get the money until the following year. By then things may have changed, priorities shifted. Or there might be something that is a little bit more topical, because of a new technique or new personnel. And I think the personnel, as you mentioned, the quality of the people, the state of their mind, the mindset, everybody needs to be probably a bit like Leonardo, be your own Leonardo to the way you approach things in general. That is something that we had discussed in terms of also the succession of people, and how do we keep quality, and how do we make sure that when you have someone else coming into the job, there's no loss of knowledge, right? Something that I think nowadays, probably more than ever, is going to be relevant, depending on funding and what happens at our centers. And I see we still have 21 participants, and it would be lovely to hear from others too, please.
Maybe one thing that we can come out from this workshop is to kind of pinpoint the promises. So if some kind of a quality management, like tracking of samples ever since from production all the way to distribution, is desired by members as it's shown right now, we can work behind the scene between 2 centers or 3 centers that are heavily implementing some aspects of LIMS or ADAPT or whatever, and collaborate with them and offer something like this in the future. Because again, as Irene was mentioning, it did come up in the think tank today as well in the survey. So this is exactly why we're having these kind of surveys or mini feedbacks, so that we can really keep track of the real desires of you people.
Yeah, from what I know in terms of, I came in with NCPN after I was not part of the initial push to, as George just mentioned, to hire someone who would design these for everybody. I think I came at a later stage, or at least I wasn't aware that I came at the stage where there was a big workshop, George, that we attended on data management, where it was a reflection with the different commodities and to see what they needed, whether something common communal was possible. And at the time, it didn't seem to be feasible. And so this is a very important topic that I think there's momentum to get started, but can easily stall. And so I think as we approach it, we just have to be mindful of what is going to be useful and helpful to the different commodities, because we have thought of a skeleton or something that is basic and that would satisfy the basic requirements, and it's just not as straightforward. And in order to give deliverable to the NCPN, we want to think about something that can actually be given to the commodities.
Maybe in the meeting, in our next proposal, if there's a desire to hire some IT guy that can create anything for some of the commodities, it could also be included in our proposal, since this is a feedback from centers, really. Why not? I mean, because that's yeah, that's exactly the idea of having the NCPN management or something, or the tier 1 listen to what the real-time desires of the centers is. And if such if we need to hire someone for all the centers to create some common thing, why not?
Fatima, let me intervene here. I think we proposed the idea. We had this discussion over and over in each crop, each plant material. We offered, and Georges can second me here, we offered to help with the support to develop similar, pipelines, software, database, but it comes with the cost. So if NCPN is willing to cover this, we don't need to hire. We have the resources in-house, and we can just cover that. It comes with a cost. So we need to cover the cost. And when you have a programmer with NCPN Center for 11, 12 years, different than having someone and learning curve. It's I believe our programmers, even if they are programmers, I have Andy worked in the tissue culture lab for a week. He was looking at meristem, looking at tubes. Carl is taking care of all these greenhouse facilities and knowing exactly how things are done. So it's just a matter of resources. So if we get the resources to cover the time, we can look into this, and we don't need to go outside. We have the resources in-house. And
Meher, just to follow up on what you're saying, definitely we don't have to invent the wheel if somebody is in-house, whether in FPS and CCPP, whatever. And the idea is, if that's why we're emphasizing over and over again for you guys to have responses, because we do have some kind of material in hand where we can go to the NCPN management and saying, really, this is the desire. It's like our evidence.
If you tell me at the
Mm.
same time, if you tell me which is your priority now, to live with what we have as a clean plant center or pay salary for the people who watering the plants, I think timing is a little bit off unless we get more money from NCPN. And let me be transparent here. We are all suffering from low and less money for our plant material that we maintain. So we need to look what's the priority. I know that this is high priority, but we are struggling to keep people employed with the Clean Plant Center these days.
Correct. So I second and I echo Maher's comment, no reason to bring someone new. Of course, the priorities is, as Maher put it, and I frame it as core activities. We need to keep the plants alive and the people going and do the basic stuff. If there is an opportunity to invest on data management activities network-wise, the investment should be either, and that of course it will depend on the individuals if they would like to pick up a new project, so they will be paid from that project, meaning sweet potato, because we had the extensive conversation earlier. If 20% of Peter's time in our case, and I forgot Maher's name, I think Wayne, you mentioned Maher, it will go to design something for sweet potato, so be it. Sweet potato will write 20% of that person in their grant. And for me, a better investment would be to hire assistants for these people. When Peter, the period of time that our programmer, our IT person was very productive, when we had resources to hire him assistants, and don't imagine anything fancy, it was computer science students. So Peter will envision, will tell them, go and do this, and then they will go and type, and Peter had time to do the complex work. So it can be as simple as that. Someone in-house, in the case of the sweet potato, it sounded like you have access to a university student and a body of students, and these students can get instructions, they can get a backbone code, whatever fits best. I suspect FPS code probably will be all applicable for you, but you can check your code and get someone that they will get guidance from our programmers to help you. And exactly, my comment earlier, same thing with Peter. He was there, he is there, he's still joining Irene when they do what we call IPPN care or introductory propagations. He works with the plants and he knows and he sees where people struggle and where is the bottlenecks, and then he goes back to his machine and he debugs and fixes the code constantly. That's why I'm thinking the second investment is more of value if there is an opportunity to make such investment after we're done executing our core mission.
Yeah, because even with the differences between crops of, one might be a perennial, a tree versus something that is not, that is something that I suspect any of the programmers that worked with one of the NCPN centers can understand if explained. But it's true that using having someone who has already worked at a clean plant center, understanding the steps of introducing of therapeutics, of distribution, diagnostics, I think really helps. And I have found most productive to work with Peter. You can sketch all the diagrams you want, and but the kind of also hands-on that you would get from the feedback, it can be done with the camera, in the greenhouse, it doesn't have to be there, but to really see and observe, and that's how we test anything, any prototype that he develops, or from time to time, we try to have that interaction where we work on a specific task together, because he can then fix what the problem is or notice certain things. And there's that constant feedback. And if you have a programmer in a company remotely somewhere else, it doesn't really have the understanding of how the plants work or the type of work or how circular it is, you're not going to get that. So that for us was most valuable. Yeah.
We tried that as well, yes. We had an outside contractor who was supposed to set up LIMS for us, and we bought their software, and they were good, and we were able to call, but they just couldn't get it. They just, now we have reached the level, and that's, yeah, we're using the software. We find something that either takes too much time or maybe can be, you know, the other way. And all we do is a quick text to Peter, Peter, this part, can you link this with that? Or can you, and then in 2-3 weeks it's done. It's linked, the data management conversation is linked, and oh my God, now I'm stuck. Scott brought it up in his presentation, in the video presentation. What these systems do is they free the time. You know, you have time and money in the axis, right? If you if you can free time for people, you save money, because now the people, Catherine, she don't have to sit down and retype everything, right? She will have time to do something more valuable, because now the PCR machine uploads the data directly into the system, not print out a paper that needs to be retyped or scanned or whatever. So at the end of the day, data management allows people to do the core function of the program more efficiently and more economically.
I don't want to interrupt your conversation, but I'm going to drop off. But before then, I just wanted to say that I really appreciated hearing from all the different labs and all the different things that are going on across the network. And I think there's, this kind of workshop is a great opportunity to see what other labs are doing and pull all the best ideas from the network and see how you can adapt them to your own laboratory. So anyway, Irene and Fatima, I just wanted to thank you for bringing this together.
Thank you. Thank you. We will be in touch soon for the next steps. Exciting. Thank you.
Yeah, I hate to be rude too, but I kind of have to go as well.
No,
but
you're not
thank you
at all.
very much.
Thank you. We should probably close it. Yes, it's 12:37, but we've had a good conversation. Thank you very much. Please, please, please do submit the Google form. Will be most appreciated. We'll be circulating the link by email again, but if not, if you have it, save it and send us feedback so we can document that properly and prove it that we got that kind of feedback. Okay.
Thank you very much.
Thank you.
Bye. Thanks.
Bye-bye. Thank you. Bye-bye.
Thank you.
Bye-bye.
Thank you.
I will. Okay, I will stop the recording now.